Welcome back. Today's study tip. After reading a page in the book, close it. Then, from memory, write a few sentences and keywords about what you read. What do you remember and what do you understand? Okay, today is cell structure and enzymes, chapters 3 and 4. So you're in for another long lecture today. Some of our more focused physiology lectures will actually be shorter. So it all evens out in the end when we get some lectures that are long and some that are shorter. Okay, so hang in there. All right, so you are probably familiar with the cell. Of course, we're focused on the animal cell, which does not have a cell wall. But all cells, all types of cells, have to have a cell membrane. That's bare minimum. Okay, so like I said, we'll focus on animal cells. And so when we focus on the animal cell, we need to learn the different organelles, right? So the first thing is the fluid portion of the cell called the cytoplasm or cytosol. Technically, cytoplasm refers to, I think, plant cells, and cytosol is for animal cells, but everyone knows the term cytoplasm, and so it's just so easy to use it. So we will say, for the purposes of our class, that you can use them interchangeably. If you go into upper division courses, then they'll probably be more strict about getting that correct. Okay. But in either case, the cytosol is the gelatinous, kind of gel-like aqueous mass or substance that's inside the cell, and all the other parts of the cell are suspended within it. So it suspends proteins, organelles. This is the matrix or the substance for all the chemical reactions to occur in. If you were taking a chemistry class, you would use usually something like water in a beaker, and then you would add your chemicals to it and you would have your chemical reactions take place like that. So it's similar. In the cell, it's a gelatinous, but it's still a aqueous or water-based substance for chemical reactions. Okay, moving on. Some cells contain fat or glycogen within their cytoplasm as well. All right, cytoskeleton is used for the support, shape, structure of the cell, and so it helps keep things in place. So the cytoskeleton is made up of proteins, and just like how our skeleton is made up of bones, right? The cell's cytoskeleton is made up of proteins, and that helps keep organelles in a particular place, more or less. Okay, and helps give the structure to the cell. And I think that's all I wanted to talk about on this slide. Okay, so cytoskeleton, right, that I was just talking about, is this mesh or scaffolding that you see here. All these, they actually in this image kind of look like bones, right? So that kind of helps you to realize that that's the cytoskeleton. And then you can see that there are all sorts of substances kind of trapped within this matrix, right? So we have the mitochondria, and see how it's kind of caught in this web, the cytoskeleton. Same thing with the endoplasmic reticulum. We have little proteins and polysomes, and so now here we're starting to see, okay, what is the cytoskeleton? What actually is it? And I told you that it's protein, and now here we see that there are microtubules. So the inside of this is hollow. It's a tube. Okay. You might have been wondering, what is a polysome, right? So a polysome is a ribosome with the RNA, the mRNA, attached to it. Okay, so it's a complex of mRNA and 2 or more ribosomes that are actively translating the mRNA into polypeptides. Okay, is everything else here you're probably familiar with. So a little bit more about this cytoskeleton. Now, this is a really nice image using a fluorescent dye to mark specific type of protein so that you can visualize it under a fluorescent microscope. I did a lot of fluorescent microscopy for my graduate research with dengue virus. Okay, so very useful tool. All right, back to our topic, cytoskeleton. So the cytoskeleton is made up of microtubules, those hollow, it's like hollow bones, right? They're proteins though, and these are the largest of the cytoskeleton structure. And then we'll also see microtubules when you look at cilia and flagella. Now, of course, this flagella is referring to sperm because in humans, right, or animal cells, we only see flagella when it comes to sperm. None of the other cells of the body would have flagella. When you talk about bacterial flagella, they're a lot more simple and they are not made of microtubules. All right, then you have smaller components called microfilaments. And so they're even smaller than microtubules. And this still is giving structure and support. And so now we'll see things like microvilli of the small intestine will have a lot of microfilaments. And actin and myosin, which you're probably familiar, that give muscle its function, its ability to contract, they are also microfilaments. Okay, so you'll see actin and myosin later in this course when we study muscles. All right, so this is for you to practice, making sure that you still remember all the parts of the cell. If you've forgotten, since I don't know when the last time you took biology was, how often are you thinking about the parts of cells? So this is a blank one for you to practice. I suggest you print a few of these out. The first time you might find yourself needing to reference the answer key, but then you want to be able to do it strictly from memory without looking at anything. Then once you're able to do that, I suggest you take it a step further and write out the function or definition of each of these organelles. Okay, so that's one of your homework assignments that you're not turning in. It's just for you to study. And then of course here's the answers, right? So be able to identify if I point to a part of the cell, be able to identify what the organelle is and what the function is. I could sit here and I could read to you each and every single one of these organelles and their functions, but you have this available to you in your textbook, and I'm sure no one wants to listen to me that badly. Okay, so plasma membrane. As you know, all cells have to have a cell membrane called the plasma membrane, and it's made up of what we call a lipid bilayer, two rows of lipids. And notice that the fatty acid tails are together in the middle, and the phosphate heads are on the outsides, and that is because of the properties. Fatty acid tails are hydrophobic. They don't like water, so they're hiding together in the middle. The phosphate heads, on the other hand, are water loving, right? They're hydrophilic. And so there's going to be the aqueous environment inside the cell, and there's going to be an aqueous environment outside of the cell. And so it makes sense then, the arrangement of these phospholipids. The bright yellow spots are cholesterol. You only see this in animal cells, which is the focus. That's what we're talking about. Now, what's going on here with all these weird looking things? Okay, these are proteins. This protein, this huge protein here, It's not labeled what specific protein it is, but you can guess that this is huge. This is probably going to allow the transport of something from the inside to the outside, or from the outside to the inside, or it's going to be an enzyme that carries out some enzymatic function, right? Because it's huge. Whereas you have these skinny, long filaments, and these are probably acting as like a messenger system, right? Something can bind here, and then it lets the inside know without transporting it into the cell. Okay, there's all sorts of different functions of surface proteins that we'll see throughout the class. Okay, these green stacks of it says Glycol Lipid, so glyco for sugar or glucose, lipid for fat, right? So this is a combination of different types of molecules to make a functional unit. Okay, you have a glycoprotein, which is just a sugar and a protein combined, right? So you have all sorts of different carbon. Here you have just carbohydrate, and of course they all look the same. They're just green blobs because this is just a cartoon picture, right? It's not an actual image of a membrane. All right, so fluid mosaic model. What does that mean? This, what you see here, looks like it's permanently structured like this, but if you were to observe an actual membrane, you would see that it's constantly moving. It's flexible. The proteins within it are able to move around, so we call it fluid because it's not static, it's moving. Mosaic because you always have all these different components in different arrangements. Okay, so I have a video. It's short. I want to go ahead and watch it. I haven't attempted to record one during a lecture before, so let's see how this works. Bear with me. Okay. So I think you can see that I'm making it as big as I can within the frame of our recording, and that's going to cut off the very bottom. So I don't think I can make it quite as big as our our recording frame, but I think this will be good enough. And you can always put in that link and watch it on your own as well. Okay, so here we go.
Living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
All right. Okay, so you can definitely use that link, copy paste it into your web browser, and rewatch that video, but that explains everything that I had just said about the fluid mosaic model of a plasma membrane. It is not static, it's in motion, it moves, right? And mosaic for all these different proteins, carbohydrates, and lipids that are within it. All right. So I already talked to you about this arrangement of fatty acid tails aggregating together to stay away from water, and then the phosphate heads will interact with water. So you get this natural arrangement. If you were to throw in a little bit of oil inside of some water, right, it's going to self-aggregate. It's going to make this, and we call these micelles. So phospholipids are when you take the phosphate and the fatty acid tails, right? So this is one phospholipid. And the head, as I mentioned, it's hydrophilic, so it will stay close to or with the water. And so the phosphate heads face outward, and that's because they love water, and they each have two fatty acid tails, which are hydrophobic, and they face inward to stay away from the water. This is another depiction of a phospholipid. This is the phosphate head, and this is the hydrocarbon fatty acid tail. This is another way to depict it, the phosphate group and then the fatty acid tails. You do not need to be able to tell me any of the chemicals, right, formulas. Just that the head is a phosphate head that will, it's attracted to water. And then the tails, there's two fatty acid tails. They're hydrophobic, so they're going to stay inward together to stay away from water. Okay? And a little bit more about the plasma membrane. Remember those bright yellow spots? I told you they're cholesterol. They're mostly hydrophobic. They will sit inside of, in the very middle of the membrane with the tails, right? And this helps give extra stability to the membrane. I think I was telling you about this in the previous lecture. So at high temperatures, cholesterol will interfere with the movement of phospholipids, making the outer part of the membrane less fluid and less permeable. So it kind of offers some extra protection. Cholesterol helps separate phospholipid tails to prevent it from becoming a hard crystal under cooler temperatures. So it's just giving more of a temperature flexibility. Then you'll also find proteins within the plasma membrane, as I pointed out, and these can be hydrophobic amino acid regions of the protein will stay inside the membrane with the fatty acid tails, and then when you have hydrophilic regions, they can go on the surface or they'll stay on the inside. And so proteins, as you know, are big and it's made up of lots and lots of amino acids. So you'll have regions that are hydrophilic and regions that are hydrophobic, and that's how you're able to get proteins that span across the plasma membrane. And then we also saw carbohydrates on the surface, and a lot of those carbohydrates are acting as cell surface markers, either for the immune system to be able to recognize that it's me, don't kill me, right? I'm not a foreigner, I'm not an invader, leave me alone, okay? Or to receive signals from hormones and things, okay? So protein synthesis is a major topic. I know there was no real segue into this. It's just suddenly now we're talking about protein synthesis. So you start in the nucleus. So this square represents an animal cell. This oval represents the nuclear envelope. So now on the inside, you're inside the nucleus. And this squiggly lines here represent the double helix of DNA. And if you open up the double helix and you copy one of the strands into an RNA transcript, then you make mRNA. Now there's actually a lot of steps involved here, but we're simplifying it down so that you remember the overall schematic. Okay, so once the mRNA has been processed, then it will leave the nucleus. And the mRNA transcript will leave the nucleus, and it has to find a ribosome, okay? And then there's a large portion and a small portion. It looks like a snowman right here. And that's the ribosome. And the string is the mRNA, right? And then with the help of tRNA, transfer RNAs, you're going to bring in amino acids in a complementary fashion along the strand, and that builds your amino acid or polypeptide, and that, once it gets folded, becomes the protein. So this is how you take a gene from DNA, make an mRNA transcript, and then a ribosome and tRNA will translate the transcript. It'll translate it into protein by bringing in amino acids. Okay, so this is a very, very generalized schematic of protein synthesis, just so you can be right. We can add on information once you have the general idea of what's happening. Okay, so DNA is always inside the nucleus. It never leaves the nucleus. mRNA is a single strand. Once it's mature, processed, it will leave the nucleus to find a ribosome. Sometimes ribosomes are free floating in the cytoplasm or cytosol, and other times you have ribosomes that are anchored in the endoplasmic reticulum. And you'll recall the endoplasmic reticulum kind of hugs the nucleus, so it's in the perfect place for this to happen. All right, so the process of taking a gene and making an mRNA transcript is called transcription. The process of taking that transcript and making protein is called translation. And so here you see a much more realistic image of the double helix, and opening it up and in a complementary base pair fashion making a copy of RNA. So remember, to do this you need an RNA polymerase to copy the DNA into RNA. And we talked about the differences between DNA and RNA in the last lecture, so you can go review that if you don't remember. Okay, so transcription is taking DNA and making RNA. But you're not doing this for the entire DNA strand. You're doing this only for one gene at a time. Okay, transcription then, as I mentioned, is taking that gene from DNA, making a transcript. So it's happening in the nucleus. RNA polymerase binds to the start or the promoter, right, the beginning of the gene. And this will cause the DNA to open up so that you can make the copy. You're going to make RNA in a complementary base pairing system. So if the DNA template has an A base pair adenine, right, then the RNA polymerase is going to put in a T. Except this is an error, because we know that RNA has a U instead of a T. So please correct this. I must have been typing very quickly. So yes, A, T, C, G, T binds with A, but when you go from DNA to RNA, T's are replaced with U, uracil. So please correct that. Okay, each terminator, once the polymerase travels through the entire gene it gets to the end, it will reach a terminator sequence, which will cause it to stop, and it will actually cause everything to disassemble. And so we say that everything falls apart, and then you're left with this strand of RNA. So this mRNA transcript has to go through several steps to become mature so that it can survive outside the nucleus without getting attacked. And so we call this maturation process. And then the mRNA transcript goes outside of the nucleus into the cytoplasm to find the ribosome for translation into protein. Okay, so one more time, make sure you correct this. When you go from DNA to RNA, you're going to match an A on DNA with a U for uracil. Okay, this will continue. You'll repeat this process of base pairing to build the transcript until you reach the terminator, and then it's going to stop, and the transcript will be it'll still be in the nucleus. It goes through several steps to become mature, and then it will leave the nucleus to find a ribosome, and then you can start the process of translation once you have a ribosome. Okay, so now we can look at translation. This is taking the mRNA transcript and making protein. So initiation complex forms, and it forms in this specific order. The small ribosomal subunit will bind with the mRNA transcript. The first T for transfer, the first tRNA arrives with the amino acid methionine. It's always this is how it always starts. Once you have your small subunit, your transcript, your transfer RNA with the methionine, then the large ribosomal subunit will bind, and now you have your whole complex. And then you will scoot down to the next codon on the transcript, and another tRNA will come in, complementary base pair. So the tRNA with the matching, what we call anticodon, will bring in its amino acid. And I'm going to show you a visual because I know this is complicated and we need to see what we're actually talking about. So right now you might be asking, what is a codon? If you forgot, a codon is a sequence of 3 bases in a row. What are the bases? A, T, C, G, but now we're talking about RNA, so it's going to be A, U, C, G. And so when any 3 in a row will make a codon. And the codon codes for a specific amino acid. Okay. The peptide bond will be formed between as each amino acid is brought into the complex. You will form a peptide bond between amino acids so that you get a chain of amino acids. Once you do the whole transcript, that chain of amino acids will then be folded into a protein. Okay. But of course, before you get a chain, you need to repeat this process over and over again. So the mRNA moves over, and then you're going to bring in another tRNA for the next codon, and you're going to continue that over and over again. Okay, so here's another video, and this will show you the process of making protein. So I do want to show this, and then I won't show any more videos. I'll leave them up to you to watch on your own, but I wanted to go over this complicated process with you. Okay, so I'm gonna end the slideshow for a moment, and I'm going to show you the DNA transcription and translation. And remember, you can rewatch this either by rewinding this lecture recording, or you can just put the URL into your web browser and watch it. It'll probably be a better quality if you watch it directly from YouTube. All right. This seems small. Let's make it a little bigger. Okay. All right.
With a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added.
So these are the steps I told you about for maturation of the transcript. I will not ask you specifically about the maturation process, but it's good for you to see this and review it because, like I said, if you take other courses, they will probably want you to know all of the information in detail. Okay, so if you're feeling overwhelmed because it's a lot of detail and processes, then I recommend that you tackle this in small chunks. You focus really hard for 5 to 10 minutes, take a 2-minute break. Maybe you watch one short part of the process, like maybe 1 or 2 steps. You review it, you take some notes, you think about it, and then you go on to the next part. You do this in chunks till you get to the end, and then you can review the whole thing together, okay? All right, so moving on.
This process is called Introns Splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract.
All right. Good job. You made it through the long video of transcription and translation. Okay, so you need to do lots of review, watching the video, writing out steps, looking in the book to master this, okay? So this is showing you just like the animation you just watched, but let's go ahead and go through it. So you have your complex, small ribosome on the bottom, big ribosomal subunit on the top. The green things are the transfer or tRNAs. They have a amino acid attached to them. They bind, and the anticodon is the green portion of the tRNA, and it will have a complementary base pairing with the codon. So we call it anticodon and codon, right? And that's how you bring in the correct and specific amino acid. And it will keep doing that. You'll keep going down and down until you get to the stop codon on the transcript. And so this shows termination of translation. So you get to that stop codon and you will have a like a tRNA come into this region, but it will not have an amino acid on it. So we call it the release factor. The release factor comes in and binds to the stop codon. That's going to cause your previous tRNA to be ejected. Your polypeptide chain that you made, which of course would be much longer than what's shown here, that's now free, and then the whole complex falls apart. This free polypeptide, like I said, it'll be lots and lots and lots of amino acids in a long chain, will then go through several steps to become folded into an actual protein, and that protein might stay inside the cell and carry out function within the cell, or it might be released outside of the cell to carry out function somewhere else. Okay? And so another schematic overall, right? You have transcription, taking DNA, copying it into mRNA. It has to become mature before the transcript can leave. Then it finds the small ribosomal subunit, the initial tRNA with methionine for the start codon. Then you get your large ribosomal subunit on top, and then you go down each codon. The tRNA comes in, binds in a complementary base fashion. The anticodon's located on the tRNA, and it matches up with the codon on the transcript. And you go down until you reach the terminator. It makes a long chain, a polypeptide chain. The polypeptide chain is made up of amino acids, right? And that's what we mean by translation. You've now gone from the language of DNA to the language of protein, right? You've gone from nucleic acid to amino acid. And then you fold it up and you get a protein. So when it's no longer overwhelming to you, hopefully you can appreciate how cool this is. Okay. Remaining chapter 3 topics, meiosis and mitosis. It's expected that you already know meiosis and mitosis before you get to this class. I know some of you learned it, some of you didn't, some of you remember it, some of you don't. So here's a video that will go over meiosis and mitosis. It's also in the custom lab manual. So you go over both of these and really understand the difference between meiosis and mitosis. This will come back up at the end of the semester when we go over the reproductive system. So you'll want to make sure that you understand this. And of course, you'll also be tested on it in this for exam 1. Okay. I also briefly mentioned characteristics of RNA and DNA in lecture 2, but I did not go over the specific steps of DNA replication. And today I went a little bit over how we make RNA from DNA. But again, you should review and know this. Here's a review of DNA replication. Please watch this. And also look in your book. Okay, so this is on you to do this on your own. Now we're going to talk about enzymes. This will not take as long as the first part of this lecture, okay? And enzymes are fun. All right, so here, this green blob, I guess, this is the enzyme. And up here, this red portion is the substrate, and the substrate binds to the enzyme in this very specific region called the active site. And it does this because it has the perfect shape to fit into the active site. And so now you see it's the substrate has bonded or docked into the active site on the enzyme, and now you have one complex here. And what an enzyme does is catalyzes a chemical reaction, which means it's going to break a bond or make a bond. And in this example, we see that it's breaking a bond, and you're going to be left with two products. Another option would have been to rearrange, and so you just get something slightly different than what you started with. This is very common in metabolism. You have lots and lots of enzymes that slowly take some substrate, and it goes into one enzyme, it comes out slightly different. Then it goes into another enzyme and it comes out a little different. And then by the time you get to the end of the entire process, you have a completely different substance than what you started with. And in fact, you'll see that with glucose and metabolism in our next lecture. Okay, so enzymes can put things together, break things apart, or rearrange things. Okay? So proteins that catalyze a reaction is the definition of an enzyme. They increase the rate of reaction. So enzymes are super cool because they are very fast. And a reaction that could happen on its own, but when you add the enzyme, it happens much, much quicker. If we didn't have enzymes, we wouldn't be able to live. It would take so long for all of our chemical reactions to happen that we would die waiting for them to happen. Their shape, the enzyme shape, determines their function. So they are highly specific to their particular substrate. And this is what we call the lock and key model. There's only one key that's going to fit into the lock. And so in this case, the lock would be the enzyme and the key would be the substrate. Okay, so the lock and key model again, right? So you could have substrate A and substrate B fit into the active site of this enzyme, and then the enzyme does a rearrangement, and now you have two products that neither of them are exactly the same as what you put in. So you put in substrate A, substrate B, the reaction occurs, and you get a product C and D in there. You can see how it rearranged the substrates. Okay? So I told you there's lots of options. Enzymes can put things together, break things apart, or rearrange things. Okay. So what do you remember? The substrate binds to the, write it down. The enzyme substrate complex forms next, and you can get a change in the arrangement of, and then the release of, and then the enzyme is free to do it all over again. Okay, so how'd you do? What did you put? The substrate binds to the active site of the enzyme, and that forms the enzyme substrate complex. Good. And when the enzyme substrate complex is formed, then the enzyme's going to change the arrangement of bonds in the substrate. So it's going to break or make bonds, and that gives you a new product, right? So you get the release of product, and the enzyme will then be free and ready to do it all over again. So here are the answers. Okay, so why enzymes are considered catalysts? How is it that they speed up these chemical reactions? So in order for a chemical reaction to occur, you need two atoms or two molecules to collide and hit each other in the perfect place, which we call orientation, right? In order for it to interact and make or break a bond. And of course that requires energy as well. And so we call that energy the activation energy, the amount of energy needed to change the configuration of the reactants. Okay, so the reaction rate is the frequency of collisions that have enough energy to bring about a reaction. And ways in chemistry class that you've made reactions happen more quickly is by like increasing the temperature. So all those atoms and molecules are moving faster, they have more energy, and more bonds will form or break. Another way to do this would be to increase the pressure in the system, or, and what we are talking about today, to have an enzyme. So let's take a look at what it looks like when an enzyme is used. So this red line represents the enzyme being present, and the blue line represents the reaction that does not have an enzyme. And so you see your initial energy level here, and A and B are the reactants that are that you're starting with. You would need to add all of this energy from here all the way up to here in order for the reaction to happen without an enzyme. And so that difference from where you started to where you have to get up to trigger the reaction is called the activation energy. Now, if you have an enzyme present, look, it's cut that activation energy way down. You still have to have some energy. Those reactants need to be able to come into contact with the enzyme's active site, right? And you need to be able to overcome the current arrangement to rearrange them. But the enzyme does a huge amount of work. And in fact, one of the main ways that the enzyme is helping these chemical reactions to happen is it's ensuring that your reactants are in the perfect place, in the perfect arrangement, right? So you don't need to have a whole bunch of collisions until one of them finally works. It's going to work on the first try because it's going to fit into the enzyme's active site. Okay, so now that you've got the basics down, let's add a little bit more complexity. Enzymes can have another component called a coenzyme. Sometimes it's called a cofactor. So the cofactor is a non-protein portion that is required to fit into the enzyme, and that gives you the rest of your active site so that the substrate can bind. So if you don't have this coenzyme or cofactor, then you won't be able to have binding between the substrate and the enzyme's active site. So once you have all of the components, then we call it the holoenzyme, which just means whole enzyme. Okay, and so cofactors are commonly metals. These are your minerals. You don't need them. You need them in very small amounts, right? And so an example would be magnesium. And then coenzymes are usually derived from your vitamins. So like, you have vitamin B, right? And we refer to the different B vitamins as like the energy vitamins. And that's because they serve as coenzymes in cellular respiration or metabolism to make ATP. So now when you take that daily vitamin, you get that boost of energy because you're getting your B vitamins, and that helps you to take the glucose that you eat or any of the food that you eat and turn it into ATP. Okay, but please be careful not to consume high doses of vitamins and minerals. You really should get all the nutrients you need from your diet. If you're eating fruits and vegetables, then you should not even need to take supplements unless a doctor has told you to do so. All right, enzymes continued. Enzymes in biology tend to have the last part of the word be ACE, A-S-E. So example, lactate dehydrogenase, abbreviated LDH, but when you see the whole word, it ends with ACE, and so it tells you, oh, lactate dehydrogenase is an enzyme. This enzyme must be working on lactose, right? Lactate, and dehydrogenase means it must be removing hydro for hydrogen. So it turns out, we're removing a hydrogen from lactic acid. And that's why it's called lactate dehydrogenase. ACE, because it's an enzyme. What is the enzyme doing? It's removing a hydrogen. Okay, so names can tell you a lot about function, right? We can diagnose a disease if we detect certain enzymes in the blood. So an example would be creatine phosphokinase. Should not be found in the blood. There are also, you can have a disease if you don't make enough of a particular enzyme that you need. And then one example, it's not really a disease, but like an easy to understand example would be someone who's lactose intolerant. They don't have the enzyme to break down lactose. And then if they eat something with lactose in it, they can have bloating, gas, right? They can have diarrhea, all sorts of fun symptoms, right? Now imagine something that's actually crucial to a process in the body. If you didn't have the enzyme that you really have to have in order for some function, then you're going to have a serious disease, right? And so that does happen. We do have cases like that. And sometimes we can figure it out and we can supplement and give them an enzyme. And other times they have to change their lifestyle to avoid consuming certain things that they can't break down, or to make sure they get something that they can't make. Okay, what else? Enzymes, in order to function as quickly as possible, need to have a particular pH and temperature, and we call this optimal pH and optimal temperature. So they can work the fastest under the optimal pH and optimal temperature. So what does work fast mean? What do we mean when we say it can work the fastest? It means it can take the substrate and release product, right? So take substrate, bind to the active site, release product. So it can do that process the quickest when you have the correct temperature and the correct pH. And so we have these graphs that demonstrate these ranges. And so here's a temperature range where the colder you get, the slower those all those substrates and enzymes are moving slower and slower. So it's going to take longer for it to get to the active site and release a product. Okay. And of course, if you get cold enough, you freeze it and it completely will stop, like hitting a pause button. Once you warm it back up, it will work faster and faster. But if you get too hot, you start to slow down and you actually start to denature. And once you denature, there's no going back, right? So I would make a dotted line here and I would put the point of no return, right? You get too hot, you denature, and it unfolds, and that's it. It's permanent. But getting too cold is fine. You can warm back up. Okay, now we have optimal pH example where if you're between pH 4 and pH 6, for this enzyme, every enzyme will be different, but for this enzyme, pH 4 and pH 6 are good. That's acidic, so you could probably guess that this enzyme is probably something that's working in the stomach, right? Where it's very acidic. But if you get too acidic, which in this case is pretty darn acidic, if you go below pH 2, you really start to change the way that the enzyme is folded. Same thing if you go above. So pH, you will denature and reach the point of no return on both sides. If you're too far out of its optimal pH, you will denature it, okay? And so here's a very important illustration of enzymes' speed, okay? So the enzymatic activity, how quickly it can release product. and its correlation to the amount of substrate present. So if you keep adding more and more and more and more substrate, your enzyme's gonna produce more and more and more product until you reach this point here. And what is this point called? The saturation point. It means every single enzyme is bound to substrate, and there's no way to get more, or there's no way to speed up the reaction. You've gotten it as quick as it can be. You've reached the saturation point. So what does this straight line mean? It means you're making lots and lots of product, and the enzymes are working as quickly as they can. So do not confuse the straight line as activity has stopped. If the enzyme were to stop, then the line would drop, right? So this is 0, and this is faster, 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 faster. This is as fast as it can go, so it's going full speed, okay? All right, we're almost done. Bioenergetics, flow of energy in living systems. So we have something called endergonic reactions. These require you to put energy into the system. Then we have exergonic reactions, and they are going to release energy. Most of your metabolism consists of exergonic reactions, and this is how you have your body heat, right? You generate a lot of heat from all the chemical reactions taking place in your body that release energy. Cellular energy is called ATP. We talked about that last time. ATP is made by an enzyme called ATP synthase. That's nice, synthesize ATP. We will talk more about this in the next lecture. But you take the precursor, ADP, and then you add one more phosphate to it, and you make ATP. So remember, ATP stands for adenine triphosphate. So ADP would be adenine diphosphate for 2, right? Then you have oxidation reduction reactions. So they're always coupled. One molecule is going to gain, and the other molecule is going to donate or lose, okay? So the molecule that gives up is said to be oxidized. The one that gains is said to be reduced, okay? And so this is an example of a redox reaction. And this is a little tricky because you have a molecule and then you'll have an enzyme. And so this molecule, it's called succinic acid, and it's going to be changed into fumaric acid. And you have this molecule called FAD, and this FAD is going to take one of the, here in blue, it's going to take 2 of these hydrogens, and it's going to become FADH2. So this molecule here is going to become reduced, and this molecule turns into fumaric acid, which has been oxidized. So fumaric acid is oxidized because you removed the 2 hydrogen, and now FADH has it. This is also important for you to know for your exam. Memorize this, understand it. It'll come up again in the next lecture. So you need to be able to tell me for redox reactions which molecule has been oxidized, which molecule has been reduced, what atoms were removed. So the hydrogen was removed from succinic acid, and it was put onto FAD to become FADH2. And when you took those hydrogen, that produced fumaric acid. All right. So that's it. Here's the answers. All right, you made it. Good job.